Today is our revised week 9. This includes content from chapters 15 and 17, and it's a remix that makes a lot more sense than the way your textbook categorized it. So chapter 15 covers brain tissue. So we'll talk about brain development, tissue organization, support and protection of the brain and structures, the cerebrum, the diencephalon, the brainstem. Those are regions of your brain regions. You have 4 brain regions. Cerebellum. Then we'll move on into motor systems in general. That's where we start with the reorganization and content from chapter 17, again, already integrated into this. And then finally, limbic system, emotional decision-making structures. And when we get to our chapter 17 exclusive content, we'll also cover memory and how memory works, which is a good thing to learn right before your final exam. So there are 4 major regions of the brain. When we say brain regions, we mean these things. The cerebrum, let me get my laser pointer moving here. This is the cerebrum, so the big meaty part of your brain that people always visualize when they think of the brain, that's cerebrum. Diencephalon is coded in blue on this image. The brain stem is coded in yellow on this image, and the cerebellum is coded in orange in this image. And all of those are the brain. You have those 4 parts of the brain. So your cerebellum is still your brain, your brainstem is still your brain. Oh, you need one of this. Your diencephalon is still your brain. Some basic vocab here. You have on your brain little sticky outy bits and then little sticky inny bits, and those are respectively known as gyri and sulci or sulci, depending on who you ask. So the gyrus sticks up, the sulcus is invaginated, is the phrase we use for that. Now, we did learn rostral and caudal at the beginning of the quarter, but we haven't seen it since. So just to remind you that in neuroanatomy, we will tend to use rostral and caudal. The reason for that is that we do a lot of neuroscience research on animals like rats, for example. And if you are a rat, then towards your nose and towards your tail, that's on one axis, and it makes a heck of a lot of sense. In humans, however, we're bipeds. And so when we go from rostral to caudal, there's actually a bend in that axis. Instead of it being rostral to caudal straight like it would be in a rat, we bend rostral to caudal. So for purposes of this class, if you choose to name things directionally with things you're more comfortable with, just say ventral, dorsal, superior, inferior, I'm okay with that in this context. Just be aware that if you go into literature, and I can also throw out rostral and caudal anytime I want. But if you're describing something positionally, feel free to use ventral, dorsal, superior, inferior. And just know that that has a bend in it. So some basic gross anatomy, surface anatomy of the brain. We do have the 4 lobes recognizable on the cerebrum. Remember, this part of the brain is just known as the cerebrum. You can find demarcations for the frontal, parietal, occipital, and temporal lobes, and they do have specific sulci between them. So this is known as the central sulcus, and that's going to be a very important anatomical feature for you for reference. For example, we're going to look at this gyrus that's immediately ventral to this sulcus, and we'll look at this gyrus that's immediately dorsal to this sulcus. This is going to be called, just for reference as we move forward, the precentral gyrus. This is going to be called the postcentral gyrus, right? That's going to be how we do naming conventions for very specific gyri of the brain. So this is a consistent sulcus. Most people will have this sulcus in that location, and it is identifiable across individuals. And most people will have this identifiable gyrus, and most people will have this identifiable gyrus. Yes? Probably, and then if they don't, it'll probably just look a little bit different or be located a little bit more ventral or dorsal or rostral caudal. It's a defined indentation. The sulcus is a defined invagination. So that one's easy. That's the central sulcus. You also have a parieto-occipital sulcus, and this is where I start ragging on your textbook because this sulcus is not identifiable on the surface the way they've illustrated it here, not in real world. There is a division between the parietal lobe and the occipital lobe. That sulcus is identifiable on a mid-sagittal section, but your textbook's lying to you in this picture. And then, but again, we can still ask you to recognize it here because there's a nice dotted line. It's just not in real life. And then the temporal lobe has this division between the temporal lobe and the frontal/parietal lobes, and that's actually going to be the lateral sulcus or lateral fissure. It's actually quite a bit deeper than your typical sulcus, so some people will refer to that as a lateral fissure rather than a lateral sulcus. You get my big brain so that I can start showing you some things. If you look carefully at this parieto-occipital sulcus right here, you can see that it's running a imaginary line across the gyrate rather than having having a sulcus right there.
I think it is brain size based on body size.
Brain size is based on body size, absolutely. As the body of an individual gets larger, the brain of an individual gets larger proportionally. And so when we measure things like intelligence, we can't go by brain size. We have to use a ratio of brain to body. And you can put other animals on that. So dolphins are actually pretty close to humans for brain to body mass. Cats are unfortunately a little bit lower, and dogs are actually a little bit higher than cats, as much as I love cats. That's what the science says, so be it. Yeah. Okay, more superficial anatomy. Cerebellum is going to be definitely caudal in this structure. And you can see part of the brainstem from the superficial anatomy view. You can see the pons and the medulla oblongata. And you are going to have to say medulla oblongata. Remember, there's more than one medulla out there. There's a lot of medullas out there. There's even more than one medulla in the brain. So you got to say medulla oblongata. You can see a couple of cranial nerves from here, and ultimately you will be able to recognize which cranial nerves are which on this image. However, this is not my favorite picture for cranial nerves. Something more like this is a better picture for cranial nerves. There's also a lab practical image where it's just cut out this area right here and zoomed in, so you can see things closer, but with less context. Oh yeah, let me give you a little bit of insight into directions for the brain. Your temporal lobe has this free sort of curved end right here. That's going to point you anterior every time. From the lateral view, from the basal view, you can also see this free curved end of the temporal lobe. That is also pointing you ventral. I have met people who made it to grad school with a neuro background without learning that, so we might as well give you that. Okay, so cranial nerves, it's in order, more or less. That's the good news. 1, 2, 3, 4. This big one with multiple splits is 5, 6, 7, and 8, 9, 10, 11, and this medial one is 12.
For what purposes can we identify cranial nerve, Roman numeral?
Right,
number?
C in Roman numeral, or cranial nerve known as optic, olfactory, oculomotor, trochlear, trigeminal. Yeah. Let's see, trigeminal, abducens, facial, vestibular cochlear. What am I doing next? Hypoglossal? Glossopharyngeal, there we go. Vagus, accessory, and hypoglossal. And then your all-important mid-sagittal section. You will ultimately be very familiar with this mid-sagittal section. This one you're going to really like. This is the corpus callosum, which does anybody want to translate into English? What's corpus? Body. What's callosum? Like a colossus? What's a colossus? Big. It just means big body. And it is a big body. We will be seeing this with more context later. This is going to be your connection between your right and left hemisphere. So this is actually cut in this mid-sagittal view. Now that means that none of the cerebrum is actually adhered. There's that fissure. There's going to be a central fissure right there that separates the right and left hemisphere, and the corpus callosum is going to connect the right hemisphere to the left hemisphere of the cerebrum. And so that's a really big identifying mark. And you can use that to contextualize a lot of other things. Your diencephalon structures are in here. We'll go into detail about what all of these do functionally, but structurally this area is the thalamus. This area that is not clearly demarcated from the thalamus, this is the hypothalamus. It has that kind of like mouth shape right there. I think of it as like a mouth shape of a seahorse going on right here. There's the eye of the seahorse in the thalamus. There's the mouth of the seahorse and the hypothalamus. And then your pineal gland, as my cursor is currently right on the pineal gland, so I'll move it away so that it's next to the pineal gland. And that's going to be in that dorsal or caudal portion of the diencephalon. You can also find it by sticking a probe in between the cerebrum and cerebellum and just going straight back, bink, and then you're touching that pineal gland. And that works on sheep as well, by the way. Now you can see that parieto-occipital sulcus. This is a true sulcus on that mid-sagittal section. Let's see, what else do I want to show you here? So your brainstem structures, there's 3 parts of your brainstem. Your midbrain includes these 2 little bulbs on the back here that we'll talk about. Your pons include that belly of the seahorse. And the medulla oblongata is going to start the tailpiece of that seahorse. See if there's anything else I want to talk about right now. Not really. We can see some ventricles here, but there will be better places for me to ask you about ventricles. Now we did talk about this one last week, gray matter versus white matter. Make sure you can locate that list we started last week. The list of things that mean cell bodies and things that mean axons, right? So you already have this on the list. Gray matter houses mostly cell bodies are there. That's where we're going to stain and find the cell bodies aligned in layers within the gray matter, and that's also going to form the cortex. So you can also put cortex on that list of things that mean cell bodies. Now, aside from the gray matter in the cortex, that's not actually the only place you're going to find gray matter in the brain. There are going to be some deep structures of gray matter that your textbook is going to refer to as cerebral nuclei. A nucleus is yet another thing that goes on your list as a collection of cell bodies. I was not joking around that this list gets ridiculous. Cerebral nuclei, and I know it's covered by that picture of a neuron right there, but it's just nucleus. And then white matter is going to be myelinated axons. It's going to have that whitish appearance because of the myelin sheath. So you will not find cell bodies in white matter. You will find myelinated axons. You will find pathways. Information is traveling through that space. So here's what that ends up looking like. You'll have that gray matter in the brain. The gray matter is going to be superficial, at least the neocortex is going to be superficial. And then the white matter is going to be deep. And then anytime you have deep gray matter, that's going to be referred to as a nucleus. And now remember from last week talking about the spinal cord. In the spinal cord, the gray matter by definition is deep, and the white matter by definition is superficial. And it's just about the direction of flow of information, right? Your gray matter, your neocortex, is going to be able to execute decisions, and that information is going to be sent out through the brainstem to the spinal cord, and then it'll be sent out centrally from the spinal cord to the periphery. So the action potential, let's say for a motor pathway, would be initiated here, direction of flow of information would be that way, and then motor neuron here initiates motor impulse and goes out that way. So it makes sense that that's where the gray versus white matter is. Does that work for everybody? Am I speaking French this early in the morning? Nope, okay. And just in case you're behind on your list, this is kind of a list of things that mean cell bodies and axons. So let's go ahead and fill out most of the rest of that list. And we started this last week. The word ganglion refers specifically to a collection of cell bodies in the peripheral nervous system. So it's not actually correct to call something a ganglion if it's within the brain. I will talk about something that is named ganglia even though it's in the brain, and that's why your textbook has given it a different name. A center or a nucleus are both going to be cell bodies in the central nervous system. We talked about nerves last week. You know that a nerve is going to be a collection of axons. It's going to be a pathway in the peripheral nervous system. Same with nerve plexus, interwoven collections of nerves. A tract is axons in the CNS. We're going to use tract and funiculus and fasciculus and pathway pretty much the same way. Those are all axons in the central nervous system. And then, of course, the cortex is that superficial layer, that's gray matter in the brain. And by the way, sometimes we refer to this as the medulla of the brain. It's deep to the cortex. Did we already cover what cortex means in this class? What's the translation of cortex? What's cortex means? Cortical. Cortical bone versus the what cavity? Medullary cavity. So the cortex translates to the bark of a tree. So like if you go out to a tree and you pull off a piece of bark, that would be called cortex. Whereas medulla just means the deep structure, right? So this is the cortex of the brain, this is the medulla of the brain. That's why you have to refer to that part of the brainstem as the medulla oblongata, because that's also the medulla of the brain. It's a deep structure of the brain. Just like the medullary cavity is a deep structure to the cortex of the bone. On this list, you also have cerebral hemispheres. We just mean that you're divided into right and left. So a cerebral hemisphere will have both cell bodies and axons, so that doesn't really go on our list that we made. How are we doing? It's just vocab so far. Not too bad? Okay. So there are some structures that support and protect the brain. The bony cranium, we already know about the bones of the cranium and how your brain is going to fit in that. I think during our bones lecture, I did take my Donovan brain and put it into the Donovan skull and show you all how the inside of the skull is a negative impression of the brain. And that is protective in nature. It's going to suspend it in the right shape. Connective tissue, your meninges. We did discuss the meninges briefly last week, but I will reiterate it. Cerebrospinal fluid, where you will talk quite a bit about CSF, and then the blood-brain barrier. You already know the cell responsible, the main cell responsible for the blood-brain barrier. What's that cell called? Astrocytes, well said. Okay, here's one image of cranial meninges. So as we go from superficial to deep, you start with your skin, then you have superficial periosteum around that cranial bone. Then you have the deep periosteum, and then you have your dura mater, your arachnoid mater, and your pia mater is specifically adhered to the surface of your brain. So you don't see a specific line for pia mater because it is on your brain. And it's going to follow the pattern of the gyri and the sulci. Now that you know what gyri and sulci are, you can point out the dura mater is going to skim over the gyri and the sulci. The arachnoid mater is going to skim over the gyri and the sulci. And you can also identify your spaces. So your epidural space would be between the meninges and the periosteum. Your subdural space would be between the dura mater and the arachnoid membrane. And then subarachnoid would be deep to this space. Okay there? Cool. Now they do, again, go in a specific order, dura, arachnoid, pia, and I think of a dapper gentleman doffing his hat, DAP, dapper. It works for my head. And here's a beautiful dissection showing you all of these layers: skin, periosteum, bone, periosteum, dura, arachnoid, pia. And again, all of this protects. That dura mater also invaginates to create a structure known as the dural septa. And there's quite a bit of anatomy here as well. In fact, we've got a really good representation of the dural septa somewhere in the room. So your dural septa are going to work about like this. What you have is the division between the two hemispheres, and invagination of the dural septa that separates those two hemispheres is known as the falx cerebri. So the dural septa separating cerebrum from cerebrum is falx cerebri. There's a bit of dural septa separating your cerebrum from your cerebellum. It's kind of like a tent over that cerebellum, and that's called tentorium cerebelli. Also embedded within these dural septa is the venous system, venous drainage from your brain. So within, and you can see it in this cross section as well. We've got our dural venous sinus is what this is called, embedded within your meninges. So it's basically a gigantic vein, and the walls of that gigantic vein are attached to your dural dura mater, dural venous sinus. So this is the superior sagittal sinus. So right along this midline, you'll have this gigantic vein right here. And then right along here, just superficial to your corpus callosum. So note how this is going to immediately surround the corpus callosum. Remember, this is going across from left to right, so it can't go any further than that, right? That's where you're going to have your inferior sagittal sinus. The inferior sagittal sinus will connect to the superior sagittal sinus via the straight sinus to this bit right back here called the confluence of sinuses. They just come together. It's a confluence. So you will be able to see these structures when you do your sheep brain dissection. Tessa.
So what's the arachnoid matter? Does it look like that in the brain, like kind of like a web? Is that,
It is very webby,
it
yes.
does look like that.
It's not, we're not going to see it very well in the sheep brains that are preserved. We'll see some arachnoid granulations or arachnoidvilli that are represented right here, but in less preserved, yes, there's like a cobwebby layer. Now that confluence of sinuses is then going to continue to drain through the sigmoid sinus and then to the jugular vein. So your jugular vein is going to finish draining that. And yes, you need to know all of that. Questions, comments? So again, what this is doing is keeping things in place. People have asked me before, what happens if you have a hemispherectomy? Are you going to like be blobbing around like a jellyfish inside of your skull? And the answer is no, you're going to fill that space with CSF, and then your dural septa are going to help keep things in place from there. You also have empty spaces in your brain. This is very, very, very vitally important. We all need this empty space in our brain. It's not entirely empty, it's filled with cerebrospinal fluid. So this helps lighten the load, it helps decrease the weight of the brain. And then really, really significant is that if there's inflammation in the brain, this is the only space this can go to. Remember that you've already got like the perfect indentation. The shape of your brain is going to be really close up to your skull. So with inflammation, it's not going to have a lot of space. If your brain increases in size, it's going to start crushing against the walls of your skull, right? We do not like crushing injuries to cells. That's a bad plan for everyone. So the fact that we have ventricles means we have a little bit of leeway for inflammation in the brain. This is also going to help with the flow of CSF, and that CSF is there instead of blood. So it's going to help nourish and control the environment, right? And that's the role of CSF anyway. So this will also be basically like your circulation of that flow of CSF. So we'll start from these two lateral ventricles. This is a three-dimensional thing. I've made these out of clay to help visualize. Okay, your lateral ventricles are in a ram's horn shape. They're three-dimensional, so they're going to start a little bit more medial in the frontal lobes, and they're going to go out like a ram's horn three-dimensionally out to the temporal lobes. That's what you're looking at. There's also going to be like a sort of horn that makes them go into the occipital lobe, so every lobe is covered by those lateral ventricles. Those lateral ventricles, by the way, just to tie this in, this is where the choroid plexus is. What kind of cell makes up the choroid plexus? Mesenchymal cells, which have what job? Making CSF. So you're actually synthesizing the cerebrospinal fluid in these mostly lateral ventricles. That's where that's happening. So this is why this is the starting place. This is where we make the CSF. Now we have these little connections between ventricles. These are the interventricular foramina, individually foramen, plural foramina. So that takes us from the lateral ventricles to the third ventricle. Now ultimately, when you've gone sort of back and forth with this understanding, you'll recognize this third ventricle is the midline structure of the diencephalon. Remember I talked about our sort of our seahorse shape, and there's the mouth and there's the eye. That should look a little bit familiar. There's actually a space between each lateral diencephalon structure, and that's your third ventricle. Come back to it later. You guys are glossed over right now, and that's okay. It takes a minute. So there's third ventricle. This is also the third ventricle. It's a midline structure. Next, we're going to go through the cerebral aqueduct. So here's the cerebral aqueduct represented here. Here's the cerebral aqueduct represented here. What's an aqueduct? A duct for water. Excellent. So if Greco-Roman, like the Grecians and the Romans invented an aqueduct system to transport water through their cities, right? Those were the aqueducts. That was infrastructure in Grecian times. And then they stopped funding their aqueducts and waste piled up and they died out. Or not died out, they got really sick and their Roman Empire collapsed. Okay, anyway, on to the fourth ventricle. You'll see the fourth ventricle is down here near the brainstem, so at the level of the pons, between the pons and the cerebellum is where the fourth ventricle is. Once you are in the fourth ventricle, cerebrospinal fluid actually has multiple pathways from here. The one that's clearest here is that it's continuous with the central canal of the spinal cord. We saw that central canal last week, or at least visually we saw that central canal. There are also some apertures. What's an aperture? Say it loud. Somebody said it, but it was really quiet. What's an aperture? Any photography people? An opening, yeah. So on your camera, your aperture can be smaller or wider, let in less light, let in more light. Helps you focus on things too. So you have 2 lateral apertures and 1 median aperture. And those apertures lead to the subarachnoid space. Now the CSF is in the subarachnoid space. It's actually going to go up and get reabsorbed into the dural venous sinuses via arachnoid granulations, sometimes called arachnoidvilli. What opening? The apertures? No, on a camera the apertures close, but on these, no, they're just open, as I understand it. Yeah, so those apertures and foramina, they shouldn't change. If they did, it would probably be a problem. These will be really clinically significant to you when we talk about hydrocephalus. Hydrocephalus could mean there's a blockage somewhere along this pathway, or it could mean a failure of reabsorption of the arachnoid villi. So again, I'm not teaching this to you arbitrarily. You will use this. So, yeah, the superior sagittal sinus is a dural venous sinus. Okay. And you do have an image specifically for that circulation. So here it is at that superior sagittal sinus being reabsorbed into arachnoid granulations. Visually in this picture, yes, right there. The lateral ventricles are also separated by a structure known as septum pellucidum. You may, if your cut on the sheep brain is very, very good, you may actually get to see some septum pellucidum. And then we'll break through it and we'll grab some choroid plexus out of those lateral ventricles. So again, role of CSF, buoyancy, protection. It's a liquid cushion. It's also a nutrient maintenance, environmental maintenance, environmental stability. Here's what the choroid plexus is going to look like. Now again, remember it's lining the lateral ventricles in a living person, but when you die it sort of sloughs off and it clumps around in your lateral ventricles like that. So here's what it would look like. So there's going to be a capillary bed on this side, and that's going to provide the fluid portion and the nutrients. And then your ependymal cells are actually basically going to take that fluid and regulate it before releasing CSF into the ventricle space. That make sense? Maybe? Okay. So the fluid of CSF is fluid from your blood. It's just going to be without blood cells. It's just going to be with a really consistent glucose level, carbon dioxide level, everything else level, right? Now here is the way your textbook is phrasing the flow of CSF. It says on your worksheet, if you just copy this worksheet, you're going to be missing some things that I care about. So my recommendation is listen to my PowerPoint and again to fill out that part of the worksheet. There's some stuff missing from this. So again, lateral ventricle, interventricular foramen, third ventricle, cerebral aqueduct, fourth ventricle, central canal or lateral or median aperture, subarachnoid space, reabsorption of the arachnoid villi into one of the dural venous sinuses. You should all just be excellent at that by the end of the quarter. Questions about flow of CSF? It's a big topic and people tend to forget about it. Sure. Lateral ventricles, interventricular foramen, third ventricle, cerebral aqueduct, fourth ventricle, central canal or lateral apertures or median aperture into subarachnoid space, reabsorption through arachnoid granulations into dural venous sinuses. we can see arachnoid granulations on dissection. They look like tiny little bumps right along that midline.
So it only gets reabsorbed if it's going through the apertures. Otherwise,
Right.
it goes
To
to
the
the central canal.
Right. Yeah, if it goes through the apertures to the subarachnoid space. Now, again, with that subarachnoid space, that is continuous with your spinal cord as well. So your spinal cord is going to get CSF through that central canal and also through that subarachnoid space. But it's only when that fluid makes it back up in this direction to the arachnoid granulations that it gets reabsorbed. Okay. Any other questions about that? I know I say it really fast, so I'm happy to repeat it many times. I've just done it a jillion times, so that's where we're at. Okay, blood-brain barrier, you already know this is astrocytes. Those astrocytes have perivascular feet. The capillary beds here, they also have a lot of tight junctions, but that's something we'll kind of come back to when we get to our blood vessels lecture in A&P2. There are a couple of places where you physiologically don't have a blood-brain barrier, places where your brain needs to detect the environment of your bloodstream. So for example, your choroid plexus, you may have noticed that there's a blood vessel right there, and there's no astrocytes with perivascular feet. It's because we're getting fluid from that. So in that location, the choroid plexus is acting as its own barrier and just controlling things its own way. Parts of the hypothalamus, there's actually a part of the brainstem as well. Again, your hypothalamus is going to have to detect your internal state. Are you hungry? Are you thirsty? Is there too much carbon dioxide? And then that pineal gland as well, which we're not going to worry about. We'll talk about the pineal gland, but not in terms of the blood-brain barrier. This is a selective barrier. It's going to keep a lot of things out. A lot of drugs are going to be kept out of the brain. If something's going to be a psychoactive substance, it's going to be something that has to, by definition, make it through the blood-brain barrier. Either we're going to take advantage of some kind of carrier protein, or it has to be, what kind of molecule can just diffuse through plasma membranes? Small and nonpolar, like our good friend caffeine. Yep. It keeps things out, except for the things it wants. So alcohol, nicotine, anesthetics, psychoactive substances can make it through the blood-brain barrier by definition. If it doesn't make it through the blood-brain barrier, it's not going to be psychoactive. And that makes it a challenge to develop new drugs for the brain. So that's what those perivascular feet actually look like. They will be pretty much continuous over capillary beds. They have to be, otherwise we wouldn't have a true barrier there. And I actually think that's a good stopping point before we get to the cerebrum. So I'm going to stop you guys here.